Faculty of Pharmacy Microbiology Department. What is gene? Genes are encoded message that tells cells how to make protein. Protein are the molecules that control the way cell behave. Cancer cells are different from normal cells. They have changes called faults or mutation. Gene faults and cancer, many gene changes that make a cell become cancerous caused by environmental or lifestyle factors. Small number of people have inherited faults genes that increase risk of particular type of the gene. What is gene therapy? Is a type of treatment which use genes to treat illnesses. Getting genes into cancer cells, they're taken into cancer cells by carrier called vector. The most common carrier used in gene therapy are viruses because they enter cells and deliver genetic material. Researchers are testing other types of carrier such as inactivated bacteria. Vectors in gene therapy are viruses. 1 retroviruses, 2 adenoviruses, 3 envelope protein pseudotyping of viral vectors. Herpes simplex virus. B. Non-virus method, 1 injection of naked DNA. 2 physical methods to enhance delivery as electroporation and gene gun and sonoporation and magnetoffection. 3 chemical methods to enhance delivery as oligonucleotides and lipoplexes and polymersomes and inorganic nanoparticles. See hybrid methods. Types of the gene therapy. Number 1, boosting the immune response. Some types of gene therapy aim to boost the body's natural ability to attack cancer cells. Our immune system has cells that recognize and kill harmful things that can cause disease, such as cancer cells. Number 2, gene therapies to make cancer treatments work better. Some gene therapies put genes into cancer cells to make the cells more sensitive to particular treatments. The aim is to make treatments, such as chemotherapy or radiotherapy, work better. Number 3, pro-drug gene therapy. Some types of gene therapy deliver genes into the cancer cells that allow the cells to change drugs from an inactive form to an active form. The inactive form of the drug is called a prodrug. Number 4, blocking processes that protect cancer cells. Some gene therapies block processes that cancer cells use to survive. For example, most cells in the body are programmed to die if their DNA is damaged beyond repair. This is called programmed cell death or apoptosis. Cancer cells block this process so they don't die, even when they are supposed to. Role of biotechnology in gene therapy. Using biotechnology methods, these molecular errors can be determined and appropriate treatments can be selected. The biotechnology techniques are explained and the advantages of each of them in the diagnosis and treatment of cancer are discussed. These techniques include gene mapping, in situ hybridization, microarray analysis and cell culture. ISH is a method for detecting RNA and DNA inside tissues and cells using labeled probes. Microarray technology provides the possibility of examining the tumor behavior in the living tissue and drug resistance of the patient. Gene mapping is a method for determining the location of genetic markers on the related genome. Advantages of gene therapy. 1. It has the ability to replace defective cells. 2. Gene therapy could offer the potential of new discoveries. 3. Gene therapy could be used in different ways to improve life. 4. Gene therapy does not just apply to human treatment options. 5. Gene therapy allows us to use technology to improve the quality of life for people. 6. Gene therapy allows us to treat the untreatable diseases. Disadvantages of gene therapy. 1. Gene therapy does not have a reliable delivery method. 2. Gene therapy is an expensive procedure. 3. Gene therapy requires ongoing treatment to be effective. 4. Gene therapy may not be able to adapt to a changing world. 5. Gene therapy might only delay the inevitable. 6. Gene therapy will shift society toward new polarization. 7. Gene therapy could change the way we think about competition. Conclusion. Patient follow-up for as much as 18 years post-gene transfer has been generally positive, with isolated tragedy. It is encouraging that many gene therapy trials for single gene and complex disorders are now complete. Vector selection and design strategies have significantly improved, and a safety profile is nearly established, as evidenced by current Phase 3 clinical trials. Though strategies such as ex vivo transduction of cells with integrating retrovirus are promising, and early success led to high hopes, it is essential to keep our expectations of gene therapy realistic. Development of targeted vectors to increase transduction efficiency and to overcome the immune and consideration of the concept of genotoxicity testing as a fundamental feature of gene therapy research. Under supervision Professor, Dr. Wael Twakol Professor, Dr. Abir Carey